This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 302, recorded on January 11, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, Ann Arbor, Michigan. That would be home of the national championship football team, my Michigan Wolverines.
Congratulations.
Thank you.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. And we're still undefeated in football. Now, granted, we've never played a game, but we're still undefeated in football. But this is the 200th anniversary of the Medical University of South Carolina's founding back in 1824.
This whole year, right?
This whole year.
Very good. And this...
Congratulations.
Is this our first TWiM of the year? I don't remember.
It is.
It is.
It's probably the second one that's going to drop.
Yeah, I think one dropped a few days ago. I don't remember. Yes, but that was recorded in 2023, which is now behind us. It's part of the history books, folks. But today we have microbiology, which is now, and we will begin with a snippet from Michele.
Yes, and the paper is entitled Staph aureus Drives Itch and Scratch-Induced Skin Damage Through a V8 Protease PAR1 Axis. It was published in Cell in November 22, and I've been itching to describe it since I saw this.
Oh, Michele.
The lead author is Liwen Dang, a postdoc in the lab of Isaac Hsu at Harvard Medical School, but it's a collaboration between 24 scientists at 8 different institutions from the U.S., Canada, and France. So the background is that Staph aureus, probably a microbe many of you have heard of, it's an opportunistic pathogen that can cause a wide range of infections, including on the skin, but also the lung and the blood. And humans are its natural reservoir. In fact, about 5% of us carry Staph aureus in our nasal cavity, and we spread it in the human population by contact, either direct skin to skin, or you might pick it up from a contaminated surface, such as at the gym, or more problematic is surfaces at the hospital, because people there are especially vulnerable. So 2 infections, for example, one is atopic dermatitis. This is a dry, itchy, inflamed skin that is a frequent habitat then of Staph aureus, and also impetigo, which is a contagious itchy skin infection that young children are vulnerable to. So here we'll learn that staph on skin can actually trigger itching by secreting a protease that clips a receptor on neurons in the upper layer of the skin that coordinate that sensation and cause us to scratch. And that behavior likely contributes to the spread of staph, either locally on that skin surface or transmitting it back into the environment, because you've now got it maybe under your nails and you can pass it to other objects or people. So this talented team identified both the bacterial protein and the host protein that interact to trigger itching and subsequent lesions. So the approach they used was really impressive. They used a combination of bacterial genetics and host genetics. And because this is a snippet, I'm going to give a higher level overview, but tell you that it was a really beautiful, logical, systematic genetic approach. So they were able to take advantage of a mouse skin infection model where they put mice into observation chambers, and the mice on their back were shaved, and then they applied the USA 300 strain of staph, which is a very common clinical isolate in this country, and it's one of the isolates that is methicillin resistant, so it causes real clinical problems because it's hard to treat. So they spread that topically, they spread the MRSA on the back of the mice and then put a patch over it, waited 5 days, and then examined the skin and also the behavior. So we see in figure 1b that it can cause what they call edema or skin scaling, thickening of the skin, or reddening of the skin. And they scored each of those pathologies and then put that together into a dermatitis score that they then quantified. And we can see that in figure 1b, where it's very clear that both male and female mice that were exposed to MRSA exhibited more of these lesions on the skin. And then to ask whether it was just the bacteria that was triggering it or whether you needed a mouse scratching behavior. In addition, they used an infrared box. So they could place the box into this, or place the mice into this box and then actually score the number of times itching behavior was observed. And let me just say, this was a lot of work. For the data that I'm going to describe very briefly at a high level, they scored the behavior of dozens of mice for each of the experiments they did. So very systematic and rigorous work. So they found indeed that itching by the mice was necessary to see these enlarging lesions. And they could say that because if they either clipped the nails of the mice or covered the exposed area with a bandage to block the scratch from damaging the skin, then they did not see the expanding lesions. So here we can show that the exposure of MRSA on the skin triggers then scratching, which causes skin damage.
So, Michele, I guess in people who have this also scratch and itch themselves, right?
That's right. Yeah, and in a number of experiments, while they use the mouse model to identify candidates, they then are careful and they systematically use human cells and human samples to verify that what they're observing in mice is also prevalent in humans.
And the scary thing is that in humans, when they begin to scratch this badly, they can develop necrotizing fasciitis caused by this staphylococci, because as they abrade their skin, it just accelerates everything and the pathology just gets out of control.
Yeah, and do you want to tell us more about that dreadful disease?
Well, it's flesh eating, so to speak. It's necrotizing, so it kills your tissue, and it's really quite awful. And both staph and strep are capable of doing this. And oftentimes it's the itch that starts the cascade, which is why this is such a significant paper.
Yeah, and it's also important to realize that Staph aureus relies on humans as their reservoir. So in order to multiply, they need to be on us. And so our behavior, that any behavior of ours that facilitates their growth or their spread, would be selected for in evolution. So having identified this behavior, they then wanted to ask what staph factors might be contributing. And here they leveraged really large detailed literature on what virulence factors are made by staph and what's known about each of them. So using a systematic approach, they determined that a regulatory system called the AGR quorum sensing system is required for the MRSA to trigger the itching behavior. So quorum sensing is a really lovely pathway that allows the bacteria to essentially count whether they're by themselves or in a crowd. And then that would, if they are in a crowd, that would flip a switch and they'll turn on particular factors that are not effective if only one bacterial cell is doing it. But if there are enough of them, they can evoke a response. So they find in figure 2b that indeed you need the AGR quorum sensing in the bacteria in order to see this host response, this itching and scratching. And then systematically knowing what virulence factors are turned on by AGR system, they took advantage of mutant bacteria that lacked one or multiple of the secreted factors. And by systematically going through all these bacterial mutants, they identified a particular protease, serine protease V8, that was not only necessary, but also sufficient. That is, if a bacterial cell lacked the gene that encoded the protease, they no longer triggered this itching to the same extent. Or if they purified this serine protease and just added that, they found a dose-dependent itching response. So having identified this serine protease, they then wanted to identify what the protease was doing to the host cells to trigger this behavior. And again, here they used a large literature that had established that there's a family of receptors in mammals called PAR receptors that coordinate different responses, including the itching response. So by taking advantage of mice that lack the gene that encodes these particular PAR receptors, they were able to identify mice, mutants, that did not exhibit the itching response, and therefore they narrowed it down to the PAR1 receptor, which is expressed on neurons that are known to be in skin, the epidermis in particular, and coordinating itching. So by that approach, they then were able to really use sophisticated measurements of looking at neuron responses to a stimuli. So for example, they could use a fluorophore that reported calcium flow, calcium flux, and they found that mouse cells that had the PAR1 receptor, when exposed to this staph V8 protease, did trigger calcium flux, and therefore the mouse or the staph V8 protease was indeed triggering activation of those neuronal cells by interacting with the PAR1 receptor. Now, they also did a number of some biochemical experiments that demonstrated that the protease secreted by staph was actually clipping part of the PAR1 receptor, and that is known to then trigger its activation. So having identified both the bacterial protease that is triggering the behavior by acting on the PAR1 receptor, they took advantage of a FDA-approved antagonist of PAR1 receptor on these neurons. And what they found is that when they treated mice through oral dosage and then exposed them to the MRSA, they exhibited less itching and also less skin damage in response to this V8 protease. So they are quite excited that they have identified not only this fascinating evolutionary fitness by Staph aureus that equips them to change the host response in a way that leads to increased damage at the site of colonization, which logically would increase spread not only locally, threateningly into deeper tissues, but probably more important for the bug is it would facilitate spread to other sites on the body and to other surfaces or to other people, and therefore allow transmission of this microbe. And they're also encouraged that there is a known inhibitor, and so people who are suffering from these itchy lesions caused by staph might be able to one day look forward to a treatment that specifically blocks this activation of the PAR1 receptor on these neurons. How about that?
It's really cool.
Yeah.
So if you get rid of the scratching, you may cut down transmission, right?
Right.
Absolutely. And if you think about it, we've been investigating, we meaning microbiologists have been investigating protease inhibitors for all sorts of things. And if we think about using a protease inhibitor applied in a topical manner, could this replace those nasty steroidal creams that are presently used to control itch in some of the more severe cases of necrotizing fasciitis to try to get people who have damaged skin from continuing to scratch themselves and expand the pathology?
So they're not treated with antimicrobials?
Well, they are indeed treated with antimicrobials, but it doesn't do anything to do the itch. I mean, because there's so many of the staph already there, and it's hard to get rid of staph off of skin because skin is many layers deep, and it's hard to deliver antimicrobials into the skin unless you're using a topical ointment.
So what would the typical treatment be? Would it be an ointment or an oral administration?
Probably both. Probably it depends on the severity of the disease.
I see.
Part of the elegance of this pathway is they did identify that it's only the epidermal cells of our skin and of mice that have the PAR1 receptor. So when they actually injected MRSA down into deeper tissue, and they did not then see the itching behavior triggered or this particular pathway triggered. So it really has evolved on the skin surface, and probably then the fitness advantage to the bacteria is spread. Well, and also replication. If you free up damaged cells and maybe release some iron and other yummy nutrients, the local bacteria could grow.
So the V8 is not required by Staph aureus?
I cannot recall. I know work has been done on it, and I don't recall off the top of my head what pathways it had been implicated in previously. Yeah. It is one of more than 10 proteases that are controlled by this AGR pathway. So
yeah, the one to ask is Michele. We did one of his staphylococcal papers, and Elio has a good blog post on the panoply of virulence factors associated with Staph aureus in one of his older Small Things Considered post that when Nizé reviewed the virulence of Staph aureus. It's Staph aureus has the Swiss Army knife that we all lust after. It's got so many blades in its pathogenesis. Its ability to cause pathogenesis. I mean, it's an incredible creature that's really been adapted for the human host.
I remember V8 because we used to buy it to do peptide maps of proteins.
Oh, nice.
I mean, years ago.
Yeah.
Because it has, it was just, I don't remember why. I don't know why.
So I want to commend these authors. Because this was a snippet, I did not have time to go into the many very rigorous systematic experiments they did. For example, they ruled out that leukocytes were involved, mastocytes were not involved, et cetera, et cetera. So they had many different mouse mutants where they could systematically check, is this pathway required? Is this cell type required? No. And then they got it, narrowed it right down to the PAR1 receptor.
So you can delete the V8 gene in Staph aureus and the bacteria will still grow. They have some growth defects. They're probably loss of fitness. So I suspect that on human skin, it wouldn't make it.
Or be out-competed by some other creature.
Yeah.
Or perhaps, or not spread as efficiently or easily.
Yeah.
It's all about fitness, folks.
You know, Michael, I've come in the last 5 years to realize that is really what it is. It's fitness.
Yeah.
It is not virulence. It is not anything else. The primary selection is fitness, and then everything else falls into place after that.
Because if you're not fit, you die.
So, for example, the reason I came to this epiphany, let's say, is that the polio vaccine strains, the OPV, have less fitness. Albert Sabin made mutants that are less fit, and there's tremendous selection in the human gut for restoration of fitness, and it happens very readily because they're just point mutations.
Yeah.
The reason they're attenuated is because they're less fit. They grow less, and they don't invade the CNS. As soon as the fitness is restored, then the virulence follows.
Comes back.
The virulence itself is not a selection factor. It's the fitness, right?
Yep.
And I would bet that is a generalizable thing, and I'm sure many people have thought of it before, but it just came to me in one of those moments. Go ahead.
We've been brainwashing medical students for years to memorize virulence factors, but it's all about fitness, folks.
I was just wondering if the same applies in bacteria because I don't have the same kind of knowledge.
Oh, yeah.
But everyone calls them virulence factors, but really they're fitness factors, right?
And really to understand the effect on the host, you can't just look at Western blots or cell culture. They needed to look at the behavior of the animal and then block certain behaviors. And again, they used dozens and dozens of mice, studying them for hours, how many times they scratched. It's really impressive.
It's very important, and it's not easy to do, as you said.
And the hallmark of this paper was the mice had the genetic lesions in their genome that actually adapted them so they could actually ask the binary experiment to know whether or not it was V8 or whether or not it was one of these other homolysins that Staph aureus has or something else. And so they beautifully displayed that, yes, folks, it's V8.
Mm-hmm.
And you almost wonder if Staph aureus is saying, my God, I could have had a V8.
Yeah.
V8 juice or a V8 car?
V8 juice. That
So
was the
as
tagline.
Michael mentioned earlier, protease inhibitors, V8 protease, of course, you should all know that a very well-known protease inhibitor is used to treat SARS-CoV-2 infections.
Absolutely.
Paxlovid, which is an inhibitor of a viral protease. Okay, same idea.
Yes.
Thank you, Michele.
My pleasure.
I have to make a title based on this paper alone. It has to be very clever.
Oh, I'm just itching to see it.
Oh, yeah. Yes.
Michael is next.
Well, you know, along those same lines, some of you may have seen the recent splash in the popular press last week. We're recording this in early January. And last week, there was a flurry activity that there may be a cause for celebration as a new antibiotic that targets the drug-resistant bacterium, which is carbapenem-resistant Acinetobacter baumannii, that often goes by the acronym of CRAB, was recently developed. And for many years, infections caused by drug-resistant strains of this bacterium have been especially a gigantic clinical challenge for the U.S. military, which during the Iraq conflict affectionately referred to those creatures as A-bomb, because of the pathology that it was causing in troops that were infected or wounded in country. And once Acinetobacter took over, it had extremely high morbidity and mortality rates. But what's remarkable is not the discovery of a new antibiotic. We've been talking about those quite often on TWiM, is the fact that the mode or the target that this antibiotic goes after is a previously unexploited metabolic pathway that specifically impacts a unique aspect of the fundamental biology of gram-negative bacteria, namely the deposition of lipopolysaccharide, the evil endotoxin molecule that we've all probably heard about. Any of you who study microbiology certainly have heard of LPS and the evils of endotoxin. And in this case, the target or metabolic path attacks the ability of the microbial cell to properly deliver this fundamental virulence factor to the surface or the outer membrane of the bacterium. So again, Acinetobacter baumannii is a gram-negative microbe. And in point of fact, the US FDA has not approved any new class of antibiotic against gram-negative microbes for well over 50 years.
50 years. That's before we had the internet, right? Before
yes.
we had email.
It sure was.
Time ago. So think of all the advances we've had in computers and
everything,
media, and yet we don't have a new antibiotic against gram-negatives.
And many of our listeners were haploid cells then.
Yeah. And this one in particular is nasty because unlike staph, which relies on humans, Acinetobacter baumannii is out in the soil. It's been found on plants. It's been found in any number of environmental settings. So we're going to be exposed to it. We need some antibiotics, especially for these carbapenem-resistant strains.
And it especially loves growing on plastic. And so if you're injured or having surgery and they have to intubate you and they put that plastic tube down your throat to help you grow, you can develop an A-bomb pneumonia for which we have no drugs.
A-bomb. That sounds pretty bad. A-bomb.
Well, that's why the military called it A-bomb.
Wow.
It literally has the consequence of a nuclear weapon.
Wow.
Now the antibiotic that these folks developed, and we'll get to introducing them here in a second, but is zosurobalpin. It's a bactericidal antibiotic, meaning that it kills the cells, and it targets a periplasmic complex named LPT, and I think the L and P stand for lipopolysaccharide, the T for transport. The B2 moiety of this multimeric protein complex lives on the cytoplasmic side of the complex, and it's an ATPase, and it facilitates the exit of the LPS moieties as they transit out of the cytoplasm into the periplasmic space, which is hydrophilic, as you know. And this molecule, or this membrane multimeric complex, is essential for delivering the LPS molecule to the bacterial surface. So the zosurobalpin acts by inhibiting the transport of LPS from the cytoplasm to the cell surface, with the consequence being the accumulation of LPS. I think this is ironic that LPS then kills the bacterium, which LPS kills people too.
Yeah, it sure
but
does.
It kills the bacterium because it accumulates to toxic levels inside the cell. Now, nature did a spectacular job. There's 2 papers that specifically discuss this antibiotic. There's a News and View article, and Nature even wrote an editorial about the need to produce more antibiotics. And all of those titles are going to be in the show notes. But this is what I just said is beautifully summarized and succinctly illustrated in the first figure of the News and Views article by Morgan Uger and Paul Hergenrather. And the cartoon nicely illustrates how the LPS complex rolls out of the cytoplasm. It's then picked up by this multimeric complex that then shuttles it to the outer membrane, where the LPS molecule just sticks out of the membrane and waves at our immune system, responsible for all the bad things that happens.
And
now,
it's amazing to me that it is a cartoon. It's so elegant, but it does not do justice to the amount of really impressive systematic work that went into identifying these down to the residues that are involved.
Yeah. Wow. And that's the chemistry paper that I'm not going to go into great detail to. And so I'll get to the drug zosurobalpin stops this process cold. LPS doesn't move once you add the zosurobalpin. If you recall your cell wall biosynthesis, and since I had to teach it last week, I did recall it. This mode of action is similar to how bacitracin, that first antibiotic we were all likely exposed to when our mothers put it on our skin knees. Bacitracin gums up the works in cell wall biosynthesis by inhibiting the production of new peptidoglycan, the cell wall of the bacterium, by limiting the dephosphorylation of this bactoprenol, which is a C55 lipid carrier. So again, antibiotics are known for their abilities to gum up the works of preventing essential cell materials from getting to their right spots. So today I'm going to walk us through one of the 2 papers that appeared in the January 4th issue of Nature. The article authored by Zamboli and 39 other colleagues. So that would take me to 4 o'clock to get through. And the second article is authored by Payhill and 12 other colleagues. And that's the one that I'm not going to touch on, but that's the one that has beautiful structural diagrams, crystallography. So I encourage you, they're both open access, so take a look at them if you have an opportunity. The Zampoloni paper.
It's also interesting to me that this is a collaboration between academic research groups and also a number of people in pharmaceutical institutes, yes?
Yes.
The Roche Innovation Center in Basel, for example.
That's exactly what I was going to say in the next sentence.
Yeah.
And the paper really has a wonderful title. It tells you what you're going to learn. This is a novel antibiotic class that targets the lipopolysaccharide transporter. The second paper by Payhill is, again, as Michele said, from the Harvard Medical School's Department of Biological Chemistry and Molecular Pharmacology and the Department of Immunology, Infectious Diseases, and Ophthalmology and Medicinal Chemistry at Roche Pharma. So this new antibiotic traps polysaccharides in its intermediate transporter, and as its title portrays, the chemistry behind the mode of action. So now, back to the Zampoli paper.
Zampoloni
paper.
It's not a Zamboni.
Zamponi. That's what I was just thinking of. So the remarkable thing about the antibiotic that they've described is that it only kills carbapenem-resistant Acinetobacter baumannii. Doesn't touch any of the other gram-negatives. This is stop the presses sort of news. This is truly a magic bullet, folks. This is great news for all the other useful gram-negatives in our microbiome because it selectively targets the thing that we want to kill most. And
so in that way, it really is next generation, because back in the day, a broad spectrum antibiotic is like, great, we can use it to treat all different things. We don't need to identify the bacterium first. Let's just treat with a broad spectrum. But now that we appreciate our microbiome is our friend and a part of us, and we don't want to disrupt our microbiome, we're appreciating anew that we don't want to use broad spectrums, not only because it raises the risk of multi-drug resistant bacteria, but also it disrupts our friends that live on us and in us.
Yep.
We're using the right tool for the right job. Not everything looks like a nail and we need to use a hammer.
Right.
And that's what most antibiotics are, is they're big sledgehammers on our microbiome. So in this first paper, Zampoloni and her colleagues walk us through how they worked this out. They examined a collection of 45,000 molecules, and these molecules are named tethered macrocyclic peptides, or they abbreviate them MCPs. And again, paraphrasing Alan Dove or Rich Condit, I don't know who uses it more often. These molecules look like chicken wire. So this is truly chicken wire chemistry that they are exploiting in order to knock out LPS transport. And if you'd like to learn more about tethered macrocyclic peptides, I encourage you to look at a blog post that I've dropped into the show notes, not authored by any of us, but it's a really nice blog post on macrocyclic peptides. Now, this class of inhibitors has an approximate molecular weight of 800 Daltons, which is larger than your typical antimicrobial or antibiotic. And as I said, the second paper by Payhill has a very informative figure illustrating how these macrocyclic peptides block LPS by transporting it, and they show precisely in the crystal structure how it actually does its deed. What they went on to show in this first paper, in this chemical analysis, is that the MCP selectively kills A-bombs or A baumannii. They write that this is a truly remarkable behavior for this class of drugs. It only does it in A-bomb. They went on to optimize the design because the MCPs that they initially tried only had marginal antimicrobial activity. And so they went on to optimize the design. Candidly, I got a bit lost in the rationale behind the chemistry optimization efficiency, but was absolutely fascinated by the experiment or test they used to assess tolerability of the drug in an animal, which was based on blood plasma compatibility. And here they offered that when they administered 6 milligrams per kilogram or 1.2 milligrams per mil of the candidate molecule to rats, the tolerability of this drug in the animal was measured by mortality in moribund animals as a consequence of a rapid decrease, greater than 40%, in lipid parameters that they measured, namely cholesterol, triglycerides, and high-density lipoproteins. So it was really bad for the animal. And this was observed after they did IV drug administration to rats, with correlated then very nicely with in vitro plasma incompatibility, which they offered resulted in the formation of aggregated low-density lipoproteins and high-density lipoprotein vesicles through yet an unknown mechanism.
I've got to give a shout out to the chemists here. It's amazing that they could see that this potential miracle drug was incredibly toxic.
Oh, yes.
And yet they could then systematically try different variants and identify one that could be tolerated.
They had to be magicians to get the MBAs in the front office to greenlight the project to do those experiments.
Audacious proposal, right?
I mean, you would think, oh, it's toxic. We can't afford any more money on this. And bottom lining this, the poor plasma compatibility is a major black mark against a lead compound getting greenlighted for further development. Yep. So what they did was they addressed this liability using, and here's where you got to give them kudos. They used a customized precipitation assay employing rat plasma, where they identified the minimum concentration of the MCPs that caused the undesired precipitation. And as a consequence of this single assay, they were able to optimize a second generation. Remember, they already looked at 45,000 molecules. They optimized a second generation of MCPs with not only activity, antimicrobial action, but tolerability, since they had this working hypothesis of how the MCPs were being toxic to the animal. They go into the incredible chemistry, which employed a zwitteronic tethered MCP, and then offered that this class of second generation MCPs showed reduced plasma precipitation compared with basic compounds, and were similar to other, and this is the underscore which probably helped them get it greenlit through the MBAs, other zwitteronic standard of care antibiotics, ones that you've all heard of, fluoroquinolones and beta-lactams.
And just to make this more accessible, a Zwitter ion is one that has both a positive and a negative charge at different places on the molecule, so they balance themselves out. So they're not overall basic or overall acidic, but rather balanced.
And so that then, this molecule had the lipophilicity. It liked to go into grease, so to speak. And they show us a couple of compound structures. They show us what the zosurobalpin actually looks like. And this ultimately helped them settle on a zwitteronic benzoic acid derivative, which they named zosurobalpin, which displayed potent in vitro activity against multi-drug resistant Acinetobacter baumannii. They then showed that the drug inhibits LPS transport, and the approach they used warrants an entire series of TWiM episodes just to do it justice.
But not today.
Not today. Briefly, they used a dynamic culture method, which means they grew many bacterial cultures, which curiously that is called a morbidostat, where I hope you can forgive the pun and visualize the assay. Where they're performing 8 complete cycles of an experimental evolution using 4 different A-bomb strains and 2 distinct growth media with or without human serum, where they gradually increase the concentration of zosurobalpin and ask the question, what's being mutated? Now then, because we can now do whole genome sequencing of single colonies, they then were able to identify 43 distinct mutations from this morbidostat, 24 of them being unique, and only 11 unique mutations identified spontaneous resistance, and 8 mutations were identified using both from the spontaneous mutations and the morbidostat.
I love that approach. Like, rather than try to deduce it from studying the literature or looking at structures, let's let the bacterial cell tell us what they need to be sensitive to this drug.
And now, since we have this power of whole genome sequencing that's affordable and quick,
right?
So where did the mutations map? Well, no surprise to any of us who read the title of the paper, it is the mutations primarily arose in the genes encoding the LPS transport in biosynthesis material.
But how exciting must that have been? Like, they screened, the whole
I would have jumped
them all to map to one protein complex. I'd be like, I'm a genius. We've got it.
I would have jumped up and down.
Oh yeah.
They identified 28 total mutations in the gene encoding LPTF, which is the first protein on the periplasmic side of this multimeric structure that's involved in transiting LPS. And it's really, they then identified several different or independent experiments that span multiple strains of these carbapenem-resistant A-bomb strains that were where the altered residue was predicted to co-localize to the luminal region of LPS, which supports the contention that MCPs may affect LPS transport. They then biochemically demonstrated this using a direct assay where the zosurobalpin and a complex in a proteoliposome, and then monitoring for ATP-dependent LPX extraction from the membrane to the periplasmic component of this in vitro vesicle, they were able to show that it was interacting with LPTA, and that's where the drug is targeting, LPTA. Bottom line of this, the drug blocked LPS extraction at concentrations comparable to growth inhibitory concentrations, and that's elegantly shown in figure 3C. But what I found to be most remarkable is that when they did the same experiment using E. coli and the same drug, the zosurobalpin, it had no effect on E. coli's ability to move LPS to the surface.
It's amazing.
So they literally show that this drug is only going to work on A-bomb. And it's a beautiful, it's got a cartoon so you can figure out what they're measuring. And then it has blots so you can actually say, yeah, I believe that. I mean, it's just incredible. Now, to confirm target specificity, they used 5 A-Balali strains, and they engineered them. Again, the genetics that Michele talked about being so powerful in her staph experiments, they again employed genetics here, each one containing one of the LPTF mutations that was found to decrease susceptibility to the drug and the virulent strains of A-bomb. So it's so nice when you have mutations to study to confirm your hypotheses. So 4 out of the 5 mutations decrease the susceptibility of this A-Balali to the zosurobalpin by at least 9-fold. The next question that I'm sure the vice presidents with the checkbooks ask is, how long will this drug have a useful life in a formulary? This is what hospitals use to say, okay, we're going to buy this drug, which is a crass way of asking, did they characterize resistance? Well, they did indeed do that. The spontaneous mutation frequency to the zosurobalpin range from 1 in 10 million to 1 in a billion at concentrations ranging from 4 to 16 times the minimum inhibitory concentration that inhibits the bacterium. And that's the frequency of spontaneous mutation that is acceptable to the checkbook writers. That the spontaneous mutation rate, that's what's acceptable for the current standard of care antibiotics used against Acinetobacter. As you might imagine, the next question is, does this drug work in vivo? To assess the potential of this, they looked at severe invasive strains of crab And they first asked about in vitro activity, and they looked at 129 human clinical isolates.
129. Let's just emphasize that.
Yes. Where do you
Wow.
get 129 clinical
Well, unfortunately,
isolates?
they're all too common, but
Yes.
to have the patience to be that
And they
systematic.
did it in vitro first. And this panel, as I alluded to, or Michele exclaimed, was biased, or should I say enriched for difficult to treat isolates. 78% of the isolates, or 32, were MDR, or excuse me, 80% of the isolates were multi-drug resistant. And the MIC required to inhibit the growth of 90% of the isolates was 1 milligram per liter, which has the MIC 90 of between 0.016 to 0.4 milligrams per liter. So for the PharmDs listening, they then characterized the pharmacokinetic properties of this drug in both single dose and multi-dose pharmacokinetic studies in mice, revealing, again, acceptable plasma exposure levels after either subcutaneous administration, where they found high clearance rates and a low volume of distribution and short terminal half-lives and moderate protein binding fractions. But more importantly, the drug lacked off-target activities in a 50 receptor panel, and the biotransformation studies found no substantial interactions with this new molecule with the cytochrome P450 system, neither as an inhibitor nor as an inducer. But again, what about the animal model? The drug was tested for efficacy in a neutropenic mouse. So effectively, these mouse have no white blood cells. They looked at pneumonia, which ABOM causes a wicked pneumonia in humans as well as mice. And using a pan-drug resistant contemporary clinical isolate, and this drug treatment of the mouse resulted in a dose-dependent decrease in bacterial burden, achieving a greater than 5 log reduction in the CFUs at the upper total daily dose of 360 mg per kg per day. And efficacy in vitro was then further characterized in a neutropenic mouse thigh model infection and an immunocompetent mouse intraperitoneal induced sepsis model. And again, ABOM causes all of those things and more in people. So again, they're doing their homework, asking if the drug is going to have broad utility. In-time kill kinetic studies, again, the drug tested against the same pan-resistant isolate used in the pneumonia model, confirmed a bactericidal effect above the 4x MIC. And again, they have beautiful graphs. So folks, we got ourselves a new candidate drug that can target pan-resistant Acinetobacter using a new target that we've never had available before. It was through their
it's gone through a couple of phase 1 clinical trials as well.
Yeah. Yep.
So it's moving.
But I think the most important or the very cool thing for me, and they referred to this cleverly as a bespoke serum precipitation assay. And it was only through that bespoke serum precipitation assay that allowed them to go from 45,000 candidate molecules, optimize it to get the development of this clinical candidate molecule. This candidate has favorable non-clinical pharmacokinetic and safety profiles. It's got demonstrated in vivo efficacy in multiple mouse models, including sepsis, thigh and lung infection. And all of these data collectively demonstrate the potential of zosurobalpin as an antibiotic. However, let's not pop the cork on the champagne yet. With nearly all antibiotics, mutations often in the target arise that lead to antibiotic resistance. That's a truism. Indeed, mutations in genes encoding components of this LPS transport system identified in the lab resulted in a striking decrease in the drug's ability to kill the ABOM. And in fact, one single nucleotide mutation in the sequence encoding the complex resulted in a more than 256-fold decrease in antibiotic activity. But the usual mode of action, and here's where the chemists and the microbiologists and everyone gets together, usual mode of action of this drug requires LPS. And this foreshadowed another potential limitation of this candidate molecule as being a good drug, because A-bomb is unusual in that the microbe does not need LPS to be viable. This bacterium can halt LPS if necessary for survival. But then take us back to the beginning when Vincent is talking about fitness. If you're not making LPS, are you going to be fit enough to cause an infection in a host?
It would clearly reduce their versatility, yes?
Yes.
Yeah.
So we're ending on a high note.
Good.
We have a new target to explore, a new class of molecules. And the one thing I'd like to challenge pharma over, perhaps we might see the development of a new category of antimicrobials like these zwitteronic MCPs that target specific LPS transport systems for the especially bad actors out there like Klebsiella pneumonia, even E. coli, or even better yet, the fats that are moved into the outer membrane of the diderms mycobacteria to help us address pan-resistant tuberculosis.
Michael, having hung out with you and Michele and many microbiologists over the years, you're always lamenting there's no research into new antimicrobials. So I guess hearing this story, the answer is if you have the willpower and the desire, it can be done. It's just a matter of convincing people to give you the money to do it, right?
Yeah, and
Absolutely.
imagination.
Yeah, imagination too. But I think people have plenty of imagination. People are full of ideas. It's just, can you get the go ahead from your company? And that's what this paper shows to me, or these papers, right?
Yeah. And the Nature editorial is talking about how to fund future endeavors in antimicrobial research. And I know you've been having a running dialogue on this on TWiM over this very thing about antivirals and that we need to be investing in the discovery of new antivirals. And I think we need to, look at like the editorial in Nature goes through of, thinking about governments subscribing to this form of research funding.
For sure.
So it's interesting that the complex that takes LPS from the inner membrane to the outer membrane of bacteria itself is shared among a wide variety of bacteria, but there are subtle structural differences that conferred the specificity of this drug to abamani. So that identifies this large protein complex as a really promising target in your pathogen of choice.
And that's what the second paper goes into in great detail, how they were able to equivalently prove that the zoroastribalpin only works on A-bomb and how it only works on the A-bomb's LPT system.
Really beautiful structural biology for those of you who are structural chemists.
Well, there you go. 2 great papers to start off 2024. It can only get better, folks.
We can only try.
You can find the show notes for TWiM at microbe.tv/TWiM. If you enjoy what we do on this and all our other podcasts, we'd love to have your support. It doesn't take very much a month. You can go to microbe.tv/contribute. And if you have any questions or comments or suggestions for papers, Mark Martin is always sending us suggestions for papers.
Oh, yes.
You can too. TWiM at microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you. Pleasure to be with you.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.